The overall goals of this protocol are to examine inflammasome activation at the single-cell level via fluorescence microscopy and to measure lysis during pyrotosis by a lactate dehydrogenase, or LDH, release assay.
This method can help answer key questions in the inflammasome field, such as how is caspase-1 activation regulated. The main advantage of these techniques is that they allow the user to examine multiple steps of the inflammasome activation and pyrotosis pathways. Demonstrating the procedure will be Andreas and Hartigh, a research scientist from my laboratory.
Begin by replacing the medium from LPS-primed bone marrow-derived macrophages seated on glass cover slips with 290 microliters of DMEM5 medium supplemented with 5 micromolar nigiricin and 5 millimolar glycine per well. Return the 24-well plate to the cell culture incubator for 60 minutes at 37 degrees Celsius and 5% carbon dioxide, adding 10 microliters of 30x FAM-YVAD FMK after the first 15 minutes. At the end of the incubation, wash the cells with 1 milliliter of cold PBS per well 3 times for 5 minutes per wash, and fix the cells with 250 microliters of 2% paraformaldehyde per well for 30 minutes on ice protected from light, labeling the cells with an appropriate fluorescence nuclear dye during the last 5 minutes. At the end of the incubation, wash the cells 3 times with cold PBS as just demonstrated, and load each slide with 7 microliters of anti-fade mounting medium. Place a cover slip onto